So we finally know what causes morning sickness. It really is a spectrum. Most people have some nausea and vomiting in pregnancy, but at the very end of that clinical spectrum are people who cannot eat or drink anything, and they are diagnosed with hyperemesis gravidarum, or HG. And HG can be life-threatening. It's believed to have killed the famous author Charlotte Bronte. It was recently reported to be the 4th leading cause of maternal death in Botswana. And patients have died in the US and the UK, mostly from severe electrolyte disturbances and severe nutritional deficiencies. And babies can die from therapeutic termination and also from fetal loss. I lost a baby at 15 weeks gestation from HG, and that is why I started studying it 20 years ago. So contrary to popular belief, there is evidence for gestational appetite loss in the animal kingdom. In placental mammals, they usually start having nausea and vomiting around week 3. In dogs, it can be severe enough to require pregnancy termination. Birds and reptiles also have appetite loss in pregnancy. There's even a study in snakes that shows that if you put a mouse right in front of their face, some snakes will not even eat that during the entire 2 months. The most extreme example is in the octopus. If you've seen My Octopus Teacher, or if you haven't seen it, I highly recommend it. It is an incredible documentary that shows how these very extreme behaviors have evolved to avoid predation by sharks. And one of those extreme behaviors is that the mother cares for her eggs without feeding until she dies. And so it is likely a biological behavior that has evolved, that is coded for by our genes, and in some species, the benefit of complete starvation until death outweighs the risk of a hostile environment. And in this case, it's a hostile environment from predators, whereas the original theory has always been that it is evolved to avoid teratogens. So I partnered with the personal genetics company 23andMe, and we did a GWAS and found 2 genes, and very fascinatingly, one of those 2 genes turns out to be the human homologue of the gene that is associated with this death spiral in the maternal octopus. So those behaviors cross from humans all the way to octopi. The other gene is called GDF15, and GDF15 also showed up in my whole exome sequencing study that I did with the Her Foundation and Regeneron. And in that study, the only variant that occurred in more than 10 cases and none of my controls was a highly pathogenic mutation in GDF15. And interestingly, the hypothesis has always been that this is caused by the pregnancy hormone HCG, and mostly because it rises and falls in the first half of pregnancy when morning sickness is most common. But GDF15 also has a peak in the first half of pregnancy. So beyond the genetic evidence, GDF15 is a cellular stress response hormone. It activates the vomiting center of the brain and causes appetite loss, taste aversion, vomiting, and weight loss. In humans, higher GDF15 levels are associated with the placenta. So the placenta makes more GDF15 than any other healthy tissue in the body. Higher levels have also been found in patients taking antiemetics, patients that have second trimester vomiting, as well as patients hospitalized with HG. And it's also, fascinatingly, the cause of cachexia. So patients that have cachexia, which is a disease or condition that's very similar to HG, where patients have weight loss and muscle wasting and loss of appetite. 20% of cancer patients actually die from their cachexia rather than from their cancer. And it is also caused by GDF15. And then chemotherapy, nausea and vomiting is also caused by a GDF15. And so this work has really incredible clinical implications. First of all, I'm not talking about GDF15 as a weight loss drug, which is being studied now, but there have been studies in monkeys that show that in a model of chemotherapy-induced nausea and vomiting, that when you inhibit it with an antibody, you can greatly increase food intake and you can lower the bouts of vomiting. So what are the variants that I identified doing? Well, I thought that maybe when the mother has that mutation and the baby inherits the mutation, she would be more likely to get HG. But actually, although my numbers are small, it looks like the opposite is the case. And this was very confusing and surprising to me. So I partnered with two brilliant people who helped me over the last year to figure this out. And what we found is that the HG risk variants are actually associated with lower levels of circulating GDF15 prior to pregnancy. So we did an elegant experiment that showed that when mice are desensitized to an acute dose of GDF15 by low dose GDF15 pre-treatment, that can be a way to desensitize them to GDF15. And so in conclusion, HG may have evolved to avoid predators, not just teratogens. GDF15 is the most likely cause of nausea and vomiting in pregnancy and HG in mammals, and therapeutics can now be developed with two mechanisms that we've identified. One is through desensitization to prevent HG prior to pregnancy, and the other is to use inhibition during pregnancy to block GDF15 to treat HG. And many people involved